within the body, and ultimately plays this central function in regards to diverting materials to the circulatory system. One of the important things, we had a look at this one in particular, is the pyruvate carboxylase. So if we think about our system here, the pyruvate carboxylase is going to give us the capacity to bring oxaloacetate as an energy source into the phosphate pyruvate system. And so what we see here is the consumption of energy in the form of ATP to give us that capacity to utilize the condensation reaction between bicarbonate and pyruvate. So this gives us an alternative approach to producing oxaloacetate. Now, the oxaloacetate can then make the choice, depending on the cellular system, whether it goes into the TCA cycle to match the concentration coming in from the acetyl-CoA, or take that oxaloacetate and move it out of the TCA cycle and bring it into production of phosphatidylpyruvate. So we're bypassing some particular reactions in regards to maintaining this oxaloacetate concentration. And as I said in the previous lecture, the intermediates become then allosteric modulators and regulators in regards to a number of these particular enzymes. And with the pyruvate carboxylase, we're going to see any of the intermediates in regards to their concentrational capacities particularly stimulate the pyruvate carboxylase to make more oxaloacetate to compensate in the decrease. So here we have the decrease of oxaloacetate and the increase of the TCA, of any of the TCA intermediates, stimulate this enzyme to produce more oxaloacetate via pyruvate, not going through the TCA cycle with the condensation reaction and basically bringing it through fumarate and malate, but bringing it through a different system. So that oxaloacetate can then actually then go off to the TCA cycle. The acetyl-CoA, the product coming from the pyruvate dehydrogenase complex at the consumption of pyruvate, also stimulates the pyruvate carboxylase system to also increase the oxaloacetate concentration. So again, this focal point in maintaining a high concentration or a steady state concentration of oxaloacetate. But if there's any changes in the consumption of oxaloacetate, whether it be acetyl-CoA, the concentration is going to come up very, very quickly and then be shunted out to the phosphatidylpyruvate. Now, we know for a fact that this requires a molecule, a cofactor called biotin. We're going to have a look at this today in regards to the enzyme clef. So we've already got the idea where we're getting our bicarbonate from the carbonic anhydrase with water and carbon dioxide within the red cell, the erythrocyte. We are getting that bicarbonate coming out. We're going to have that condensing with pyruvate, utilizing ATP to produce oxaloacetate. Our alternative component here with the phosphatidylpyruvate carboxykinase gives us that capacity to take that oxaloacetate, utilizing importantly GTP, not ATP this time, but GTP because of the specificity of the enzyme, to allow us to basically cause a decarboxylation reaction. And that decarboxylational reaction then commits the oxaloacetate to produce the phosphatidylpyruvate, which we can then put into that particular phosphatidylpyruvate pool. But again, this is a capacity for us to get rid of a carbon moiety through the release of carbon dioxide to produce the phosphatidylpyruvate from oxaloacetate outside the TCA cycle. Okay, so we also see the situation with the phosphatidylpyruvate carboxylase. Now, this particular enzyme is reactive and allosterically modulated by fructose 1,6-bisphosphate, which is, as we know, is intermediate in the glycolytic pathway. Now, the fructose 1,6-bisphosphate has the positive effect in regards to activating the phosphatidylpyruvate carboxylase. And this is not so much seen in higher organisms, but more seen in yeasts, microorganisms, and plants. And what this does basically allow us to do is to further increase the mechanism that we just illustrated before by increasing the pyruvate concentration and by obviously consuming the dihydroxyacetone phosphate and the glyceraldehyde 3 phosphate, which is a byproduct of the glycolytic pathway of the fructose 1,6 bisphosphate, because ultimately this is going to promote the concentration of pyruvate. And in that case, we then have to have the matching stimulation to consume the pyruvate and the bicarbonate through the pyruvate carboxylase using biotin and ATP, but importantly now taking the phosphoenol pyruvate carboxylase and the carbon dioxide to give us then the reverse in regards to the oxaloacetate and the inorganic phosphate. So again, it gives us the capacity to provide more oxaloacetate from a separate system and separate regulational components. So showing basically there's a number of different pathways to produce this molecule, to keep it in the concentrations that we desire, or be able to supplement it in one way, shape, or form from various pathways, which are then independently regulated. So again, that is the thing that I wanted to really stress. So when we slow the TCA cycle, we're basically wanting to slow down the oxaloacetate production and move that oxaloacetate outside to the phosphoenol pyruvate system. So biotin, in this case, plays a very important role. Now, biotin basically is a reagent that is a vitamin. It is utilized in carboxylation reactions specifically. And what it does, it allows us to carry one carbon constituent. So it actually essentially acts within the enzyme as a prosthetic grip, similar to what we saw with hemoglobin and the heme moiety. Now, the pyruvate carboxylase actually requires four moieties of biotin to be bound to it, and it binds specifically to the epsilon amino function on the lysine in these particular pockets. And it needs four of those to bind to give it the biological activity, meaning there's an all or none principle. So you must have this particular molecule present, and we see it in the diet. Now, we can see this particular molecule, biotin, having a very specific interaction with another protein called avidin or streptavidin. Avidin comes from egg white, and streptavidin comes from streptococcus. And basically, this is a very interesting and essential tool in biochemistry. We talked about affinity chromatography. We've talked about labeling compounds in regards to adding fluorophores to them. Biotin and streptavidin's interaction gives us this capacity to make a modification on a protein and uses biotin moiety to attract a high affinity interaction between the avidin or streptavidin. The KD is 10 to the minus 15 moles, and it's considered nearly an irreversible reaction. And therefore, we can utilize this to attract molecules to each other specifically by their bioengineering. And we actually do this in my laboratory quite often, using biotin labels onto toxins and then using a larger protein, a GFP protein or a fluorescent protein, which has a avidin part attached to it and therefore becomes an attractant to that particular toxin. So there's a whole lot of things we can do with these, as I will actually show you in a lecture coming up towards the end of this semester. So the biotin also plays this particular role in regards to interacting with the enzyme and the substrate, in this case to allow us to condense the bicarbonate and pyruvate to form that additional pool of oxaloacetate. So let's have a look. In the pyruvate carboxylase, so here's our lysine with its long chain side chain group, and we're going to see then the biotin moiety in regards to its interaction with the biotin constituent. Now, what happens in the pyruvate carboxylase, we have our bicarbonate coming in, which is going to actually be utilizing ATP as the driving source as a leaving group. So in this case, we're going to see bicarbonate interact with ATP at a particular catalytic site, which is separate from the main site where we're going to see this activity, but they're in close proximity to each other. We're going to produce this carboxyphosphate, which is a great molecule leaving group to prime really the carboxylic acid or the bicarbonate component, the carbon dioxide, to actually become a great attractant in regards to attacking the lone pair of electrons in the biotin structure, which is now basically covalently interlinked to the pyruvate carboxylase. In this case, what we'll see is that phosphate group as the great leaving group. So the phosphate comes off, allows us then the carbon dioxide to actually then form a covalent bond with the biotin moiety. So now we have the carboxybiotin or biotinylated enzyme, which is now basically primed, ready for the issue of having the pyruvate come in to allow us to basically give us the situation of activating that pyruvate molecule and interacting with a double bond which is established from this leaving group in regards to now the carbon dioxide coming off and rearranging the bond on the biotin moiety to give you this double bond. This double bond is then susceptible to the incoming pyruvate molecule. We reestablish interaction with this moiety and allow then even the carbon dioxide that we had previously released to come back and now react with the pyruvate molecule, this pyruvate enolate form, and allow us basically to now consume the carbon dioxide to produce our friend the oxaloacetate. In the same process, we are regenerating the biotin side chain moiety in the enzyme cleft of the pyruvate carboxylase. So we're ready to go through the whole system again if we have the ATP and bicarbonate to produce the carboxyphosphate. So again, we can go through this time and time again and causing the rearrangement and seeing how the biotin plays an intermediate role in basically priming the carboxylic acid and priming the biotin structure to interact with the pyruvate molecule to allow to cause the condensation with the carboxylic acid with carbon dioxide and pyruvate to produce oxaloacetate. So it's a little different to what we saw in regards to producing the producing the oxaloacetate from the malate in the TCA cycle. So the role of vitamin biotin, I think it's vitamin H, I think it is basically to give us this issue in regards to underlining the first phase of interaction with the carboxylase. Now, I've shown this in a little more greater detail. I've already spoken to you about this in regards to the establishment and interactions of the biotin moiety and the carboxylic acid. And we see this in the first phase of the reaction, where bicarbonate is converted to carbon dioxide. And that carbon dioxide can be a later reactant in basically providing that additional carbon moiety in regards to the oxaloacetate. So in this case, we are consuming carbon dioxide, not producing carbon dioxide, but consuming carbon dioxide in the form of bicarbonate to give that additional carbon from the pyruvate, the three carbon moiety of pyruvate to add an additional carbon moiety to give us oxaloacetate. And again, we go through this same process as I've illustrated before, again, indicating this role of the carbon dioxide and its interaction with the biotin moiety, and basically allow us then convert the pyruvate to this enolate form by transferring protons and giving us the capacity then to further interact with the incoming pyruvate to produce the enolate form, which is then going to be the reactive form with this double bond going on to this particular carbon to then break this double bond to basically give us a protonation or carboxylic acid. So it could be protonated, but give us a carboxylic acid. So the second phase is where we see that donor be passed to the acceptor and basically release the oxaloacetate from the enzyme. So again, this is our first illustration of how we are further our second illustration of how we're building molecules now using carbon. We've seen this with the first example in regards to the acetyl-CoA. We see first off the direction of pyruvate going to acetyl-CoA, releasing carbon dioxide, giving us a 2-carbon constituent, now binding to oxaloacetate to give us the 6-carbon constituent in the form of citric acid. This is an example also now utilizing carbon dioxide to give us the capacity to add to pyruvate to make that 3-carbon constituent into a 4-carbon constituent, which is in this case oxaloacetate. Now, we see these types of environments in regards to their clustering and their association. And as I said before, when we looked at the PP1 enzyme for the control of glycogen synthesis, the active and deactive form through phosphorylation, we have a number of enzymes that are going to come together in regards to associating with each other and clustering. And what we see here is high cytosolic concentrations of these enzymes clustering together. And it could be ionic interactions, usually not covalent, but ionic interactions, which basically then give us this capacity to do substrate channeling, to move the substrate to various parts of different enzymes to do the work more efficiently without going through a situation of a dilutional process to then cause the more time course and energy requirement to move molecules to their particular substrate clefts for derivatization and modification. So when we start breaking down cells, we've got to be very careful in regards to disrupting these protein-protein interactions. And because they are sometimes very weak, we have to be very considerate in regards to we may be destroying the biological consequences, activity, and regulation of the enzymes that we are really interested in. So keep that in mind in regards to looking at how enzymes match each other in their particular concentrations and how they associate with each other. And even some of these other proteins that come in as regulator components in regards to these clusters of proteins that have clustered together to form substrate channeling. Okay, so we're now going to have a quick look at the regulation of the TCA cycle. And ideally in the TCA cycle, we want to ensure, if we take our photograph of the TCA cycle, that we would see the intermediates be basically at a steady state. And that's what we want to achieve. But if there is a flux or a change, we want to quickly adapt the TCA cycle in the sense of regulation in regards to responding appropriately to maintain that steady state. So again, it's a bit like having your sitting and idling at the traffic lights with your foot nearly on the accelerator to take off when the energy requirements are indicated by the cell. So how does the cell tell the TCA cycle to, hey, kick it into high gear? And if we think clearly about the needs of the cell in regards to energy, as well as the byproducts in the destruction of our energy carriers, including the issue of the concentrations of NADH, FADH2, ATP. Remember, they are going to be indicating that the cell is in a good state and that wants to slow down the further production of those molecules through the TCA cycle. Or in the opposite direction, when we start requiring the consumption of AMP, ADP, NAD+, coenzyme, which are all precursors which would be normally utilized and be made into energy carriers in a happy state cell. So if we have a look at this particular picture, it brings the message home and true in regards to what I've just said. So let's think about the first step with the pyruvate dehydrogenase complex, the commitment stage to taking that pyruvate from glycolysis, making acetyl-CoA, and bringing it into the TCA cycle. We would want to stop this reaction through the pyruvate dehydrogenase complex allosteric modulation by looking at how well the cell is. If there is a large amount of energy, why would we want to produce more NADH, FADH2, and GTP from the TCA cycle, where we could potentially take that pyruvate and utilize it in other components? So this is where the ATP particularly becomes an allosteric modulator to slow down the pyruvate dehydrogenase complex. We also think here we have the acetyl-CoA. So if we're producing enormous amounts of acetyl-CoA and not matching it with oxaloacetate, we're going to see the increased concentration of acetyl-CoA, which in turn will prohibit its further production by allosterically modulating the pyruvate dehydrogenase complex. So again, acetyl-CoA is also a negative regulator on the pyruvate dehydrogenase complex. NADH, we have a little bit of NADH coming from glycolysis, but the majority of the NADH is coming from the TCA cycle and also from fatty acids. We'll talk about that a little bit today. But ultimately, that NADH, if the cell is producing and has a lot of store of NADH, why make more? Therefore, NADH becomes a negative allosteric modulator on the pyruvate dehydrogenase complex. The same is true for the vice versa in regards to AMP, which is the degradative product of ATP and ADP. Remember, we can take one phosphate group or two phosphate groups as pyrophosphate to go from ATP directly to AMP, or we can take one phosphate from ATP to ADP and take another phosphate off from ADP to AMP. So keep that in mind also. But this time, these are positive allosteric modulators. They will actually bring the enzyme into compliance and make it work the way it needs to be at its Michaelis-Menten constant and at that steady state reaction at the Vmax. So remember, we're talking about our enzymology now. We also have coenzyme A and NAD+, which is again the byproduct of the reduction of the NADH, which also acts as a positive allosteric modulator. And as we look through the points of regulation, as I said, we have limited points of regulation in regards to the TCA cycle, where we have the citrate synthase, the isocitrate dehydrogenase, and the alpha ketoglutarate dehydrogenase complex, which are our key regulating components in regards to the commitment to the TCA cycle. So in the early stages with the citrate synthase, we're going to be specifically looking at the weight of the NADH, citrate itself, so again, negative feedback on the enzyme by the product, the concentration of ATP, because remember, this thing is going to be feeding ATP production. And if we don't need any more ATP, stop the synthesis of citrate by inhibiting the citrate synthase and then letting the oxaloacetate increase its concentration and go off to become into the phosphoenol pyruvate pool. But also this one has a sensitivity to succinyl-CoA, which is an intermediate of the TCA cycle after that of the alpha-ketoglutarate. So if we start seeing accumulation of this guy, it's also going to have a negative component in regards to regulating the citrate synthase. Now, ADP, again, the byproduct of degradation of ATP, has a positive allosteric modulation in regards to bringing that citrate synthase back up to its Vmax, accepting the acetyl-CoA, coordinating with high concentrations of oxaloacetate to produce more citrate, to then feed through the system to make more NADH and FADH2, as well as GTP. So again, it has a positive allosteric modulation on the enzyme. When we look at the isocitrate synthase, we have a modulation, again, due to the concentration of ATP and the concentration of ADP being a positive modulator, as we saw also with the citrate synthase. We also see alpha ketoglutarate dehydrogenase complex also having sensitivity to the product of succinyl CoA, NADH as a negative controller, but also calcium as a positive controller in regards to maybe the release of calcium coming from the endoplasmic reticulum through the IP3 receptor. And again, we're starting to make our connections into the components that we have talked about. So the regulatory high points within the TCA cycle are going to be the pyruvate to acetyl-CoA through the pyruvate dehydrogenase complex, and basically the acetyl-CoA and the oxaloacetate condensation reaction through the citrate synthase. So they're the key components really in regards to regulation. So we see allosteric regulation being really dominated in the pyruvate dehydrogenase complex by the health of the cell in regards to the acetyl-CoA concentration circulating as well as the NADH. But as we've seen also, AMP, CoASH, and NAD plus are all basically kind of components which are going to be stimulating their production of their appropriate forms of ATP and NADH and the consumption of CoASH, because they're going to be decreased as a representation in the activity of the TCA cycle. So we're going to see these guys become abundant if we need more energy, and they will be decreased as they are consumed to make their appropriate conjugates. So again, we're going to be allosterically modulating the central pyruvate dehydrogenase complex. So basically, what we can think about is we switch it on like a switch when to increase the energy demands of the cell, meaning we're producing more energy for the cell, and therefore increasing ideally the flux of acetyl-CoA and matching that with the oxaloacetate. Now we switch it down, or turn it off, when we have the available fuels for the cell to keep it happy, and this might include the issues of fatty acids, acetyl-CoA, but particularly the concentrations of ATP and ATP and NADH as a key regulator in regards to the entry into the TCA cycle. So we have these capacities to switch things on and off at various sites within the TCA cycle, but mostly they're approaching the 12 o'clock to the 5 o'clock position in regards to that particular sector of the citrate synthase, the isocitrate synthase, and the alpha ketoglutarate dehydrogenase complex. So let's have a look what we see in the pyruvate dehydrogenase complex, where phosphorylation becomes the key component. So in enzyme number 1, the pyruvate dehydrogenase component, where we're seeing pyruvate coming with the thiamine pyrophosphate, taking that functional group of the acetyl group, the 2 carbon constituent, spitting out the carbon dioxide. And again, this is the highly specific rate limiting step to prime the whole system. It is this guy here through phosphorylation which will either slow or stop the actual uptake of the pyruvate. Okay, so we're going to get no more carbon dioxide and no more consumption of pyruvate, and therefore no more movement of the hydroxyethyl group to the enzyme 2 and enzyme 3 complex in the production of acetyl-CoA. So we're decreasing the concentration of acetyl-CoA. So in this case, dephosphorylation can activate the kinase, where phosphorylation inhibits it. So again, this is an issue of allosteric modulation by the availability of ATP. So as we see increases in ATP, we see inactivation of the pyruvate dehydrogenase complex. So we're not adding more acetyl-CoA to the pool for the TCA cycle to produce the FADH2, the NADH, and the GTP, because we are meeting the energy requirements of the cell without further adding more acetyl-CoA into the system. But if that system changes in regards to we see increases in AMP, ADP, NAD plus, we are going to start switching the system on to get back to the Vmax to allow us to take the full consumption of pyruvate, producing carbon dioxide, bring that acetyl group, those two carbon constituents through, condensing it with the via the via the thar ester bond with a CoAsh consumption and CoA being a part of then the acetyl CoA and then feeding that into the TCA cycle. So again, decreasing ATP concentrations, decreases have the dephosphorylation and activates this particular kinase to do its job more efficiently, moving it to the Michaelis-Menten maxima and the Vmax of the enzyme function. So again, keep in mind, we are discussing this issue of enzymes. They follow all the same issues in regards to the K-1, the K1. They will have different allosteric modulators and inhibitors. So we can have, competitive inhibitors, non-competitive inhibition at different sites within this particular enzyme. So again, we can control an enzyme, particularly if it's in a complex system like this, by targeting different parts of the enzyme. Okay, so again, allosteric modulation becomes an important role and a highly specific player in regards to maybe regulating E2 instead of regulating E1. But in this case, most of our regulation is through the phosphorylation of a serine moiety in E1 in the pyruvate dehydrogenase part of the pyruvate dehydrogenase complex enzyme. And that's why it's called a complex, because it has three individual components that have all their own individual regulational components and sensitivities to the cellular environment. Okay, let's go forward. So we have these highly exogonic steps, as we've talked about in before, which are these rate limiting steps. So again, we think about our kinetic process with enzymes with the K-1 and the K1. We can actually change those in regards to how we feed the particular pathway. And this is going to depend specifically on the substrate availability. So you think about that reaction of the citrate synthase is going to depend on the substrate suitability of the acetyl-CoA and the oxaloacetate. The isocitrate to go into alpha-ketoglutarate is going to depend on the concentration of isocitrate availability going through the isocitrate dehydrogenase reaction. And same with the alpha-ketoglutarate. The concentration of alpha-ketoglutarate to produce more succinyl-CoA is going to depend on the concentration of alpha-ketoglutarate. You cannot make these molecules if they're not being their precursors are not being delivered to the enzyme at the next stage. And as I said before, the substrate of one enzyme becomes a product. That product becomes a substrate for another enzyme further down the pathway. So we basically want to inhibit the capacity of these enzymes being sensitive to the products that they produce. And as we've seen throughout this class, when we get that negative feedback, it could be very distant from a product further down the chain of metabolism. But in most very reactive and sensitive allosteric modulation, to inhibit that enzyme, to give it into that locked complex, is basically allosteric modulation through the substrate product. So the product that it produces has the capacity to give it negative feedback very, very quickly, particularly if that accumulation of that substrate or that product, so the product becomes a substrate, but let's say the further enzyme down the pathway doesn't activate it, that intermediate constituent is going to start to increase its concentration. The intermediate is then going to inhibit its own production by impacting the enzyme that originally produced it. So we have this allosteric feedback and inhibition in regards to the early stages in metabolism. And these, as we've seen, focused on the citrate synthase, the isocitrate dehydrogenase, and the alpha-ketoglutarate dehydrogenase system. We've already seen how the citrate synthase itself is required, that matching component between the acetyl-CoA and the oxaloacetate, and their availability to form then that citrate. So again, that's a key regulator in regards to keeping that hub going, because if you start screwing up with either the concentration of the oxaloacetate or that of the acetyl-CoA, we're going to change the intermediates of the TCA cycle. The other thing is we've got to be very careful is the product accumulation. If we have a enzyme further down the pathway that is not working efficiently at its Vmax for some reason, whether it's allosterically modulated or there's potentially a problem with the particular enzyme with cofactors not getting there, we're going to start seeing an accumulation of certain materials. And again, this is where we see succinyl CoA having the capacity to both inhibit the first stage in the form of the alpha ketoglutarate dehydrogenase to slow down its further production because it's all bulking up, but also further on the metabolic pathway, further up in its production, in inhibiting the citrate synthase to stop then ideally the consumption of oxaloacetate, as well as the bringing in and consumption of acetyl-CoA to give us that minimizational effect to even further stop the production of citrate, isocitrate, and alpha ketoglutarate, which ultimately will then affect and impact the concentration of succinyl CoA. So again, a very fine regulating component. The regulators could be at very close proximity to the enzymes that are there, the direct enzyme that's producing them, or it could be at an enzyme further up the pathway, which leads to the synthesis of that particular compound. So again, when we look at allosteric modulation, changing it with non-competitive inhibition in most cases, we're going to be able to then cause the regulation and fine-tuning of the kinetics of these particular enzymes to match the needs of the cell in regards to the energy requirements. And again, these are all finely regulated and attuned to the needs of the cell. So we have to have this consistent feedback in regards to signaling to the pathway in regards to whether you're a happy cell or a sad cell. Happy cell has lots of energy. A sad cell has a depletion of energy and needs to have a backup of new energy coming in. So again, when we think about this also, even outside the TCA cycle, we have another integrational component, which is either going to further stop the production of pyruvate and ideally the production of acetyl-CoA in regards to how citrate itself, an intermediate from the TCA cycle, has a major regulatory component in interfering with our friend, the phosphofructokinase number 1. And in this case, we have the capacity to then slow down the pyruvate production by inhibiting glycolysis, which will ultimately inhibit or decline the concentration of acetyl-CoA. Again, that particular pathway is also regulated by the concentration of NADH and ATP. But citrate itself, as an intermediate from the TCA cycle, now is the intermediate between those two metabolic pathways in the form of the TCA and the glycolytic pathway. If there's too much citrate and not being moved through the TCA cycle and there's accumulation of that citrate will diffuse to the glycolytic pathway, targeting the phosphofructokinase number 1 enzyme and basically slow the production of the glycolytic production of pyruvate. So again, these multiple step dependent and distant regulation coming from intermediates of different pathways now causing a structural regulation in regards to the chain of command or the chain of feed or the chain of supply in regards to these intermediates. So we're now going to switch gears from regulation to introduce additional metabolic pathway. Now, if you know the TCA cycle, this is going to be a breeze because you only have to know one particular additional compound called glyoxylate. Now, the glyoxylate cycle is pretty unique. It's not found in higher organisms except for vertebrates. Hang on. Is it vertebrates or invertebrates? Reptiles. Yeah, invertebrates. So we see this in reptiles, in bacteria, and in plants. And it gives them the capacity, particularly in bacteria, and I'll focus on bacteria, in regards to the capacity to take acetate, which is a molecule which can be easily derived, to produce and combine multiple acetate molecules to produce carbohydrates. And this is very useful in metabolism within these organisms where they're utilizing fatty acids and amino acids, because in some cases they don't have the capacity to intake carbohydrates or metabolize carbohydrates specifically in regards to dealing with complex sugars like glycogen and starches. So what they actually do, they really heavily rely on the amino acids and the fatty acids as an energy source to produce glucose. So for us as uh, vertebrates, we don't have this capacity to take fatty acids, specifically fatty acids, directly and acetate ions molecules to produce carbohydrates. So we don't have this particular system. It would be amazing if we actually bioengineered this into our systems, because it would actually give us enormous capacity to even advance our own energy requirements. Who knows what will happen in the future? So in this particular case, we see the lipid bodies, and we've talked about these very large chyromicron fatty acid containing components with the triacylglycerols with appropriate phospholipases breaking down those triacylglycerols to the free long chain fatty acids and then degrading those long chain fatty acids to become acetyl CoA. And that's the key component, the key differentiation, taking those fatty acids and directly going to acetyl CoA and allowing them to then be utilized as that uh, acetate form to then produce carbohydrates. Now, some of these reactions of this TCA cycle are basically utilized within this system, but they remove two important ones, and that's the decarboxylation. So, where we see the removal of carbon dioxide from the isocitrate and from the alpha ketoglutarate, we don't see that here in this particular metabolic system. And what the plants are trying to do and the bacteria are trying to do is enhance their capacity to utilize carbon backbones as a form of biosynthesis to allow them to produce carbohydrates. And remember, carbohydrates are a key component in cellular structure for plants in the form of cellulose. So, they're not using that as an energy source, they're using it as a structural component. So they've got to be able to produce enormous amounts of these carbohydrates to give them the structure form that they need. So plants that are rich in oils, so if you think about nuts, sunflower seeds, castor beans, and even cucumbers are actually very rich in fine oils. And these particular oils are the ones they utilize in the glyoxylate cycle. So let's have a look at the glyoxylate cycle. Now, as I said, if you know the TCA cycle, you already know the glyoxylate cycle. But the interesting thing that differentiates this cycle from the TCA cycle is that it consumes two moieties of acetyl CoA. So two constituents of two carbon moieties come into this particular cycle. So we bypass two decarboxylation reactions. We have two acetyl CoAs coming in to basically form oxaloacetate, as well as releasing a molecule of succinate. And that succinate becomes a very important intermediate in plant metabolism and microorganism metabolism to allow us to then produce carbohydrates. So let's have a look at the cycle. So the glyoxylate cycle has two differences in regards to it uses an isocitrate lysase that takes isocitrate directly to succinate. Now, it would be amazing if we could insert this enzyme in ourselves or in higher organisms, because we would then remove the decarboxylational reaction. We also have one other particular reaction, a malate synthase, where we're taking an additional molecule of acetyl CoA, adding it to that succinate to produce malate, and then going through the normal system of allowing us to produce oxaloacetate, which can then go to become phosphoenolpyruvate through the kinase and allow us to then produce more carbohydrates. Because remember, again, plants are motivated to produce glucose because they need that for their structural components. So in this case, let's have a look. We have acetyl CoA coming normally in with the citrate synthase, condensing with oxaloacetate to produce citrate. We have the anaconase in regards to its taking the citrate molecule and producing isocitrate. Now, this is where it deviates. We no longer have that decarboxylational reaction with the alpha ketoglutarate dehydrogenase complex, but we have this isocitrate lyase, which basically cleaves off this molecule here to form succinate. So one succinate molecule will leave, leaving 4 carbons from the system and retaining, in this case, glyoxylate. So again, we get a cleavage here. We basically form 2 double bonds with a carboxylic acid and a double bond to form the molecule glyoxylate, with the expense of removing that succinate. Now, that particular glyoxylate is further condensed with another molecule, the second molecule of acetyl-CoA, and that is coordinated with the malate synthase. That process produces the intermediate malate, which we already know in our TCA cycle, and that can go through the malate dehydrogenase reaction to give us one moiety of NADH to produce then our oxaloacetate. So again, it's a different deviation, an evolutional branch of the TCA cycle to give us succinate as a product, consuming 2 molecules of acetyl-CoA, producing 1 molecule of NADH by the key intermediate of this compound of glyoxylate. So again, it's an alternative pathway utilized by lower organisms to allow us to produce complex carbohydrates in the form of cellulose, moving that off, moving this guy off to be able to produce a 4-carbon constituent. So I'm now going to show another picture which shows its integration now within the TCA cycle, because these 2 cycles actually work in unison. And what we actually see here is the succinate being a role player, coming into then also feeding the TCA cycle to get that 7 o'clock till 12 o'clock position, going through the succinate to the fumarate to the malate to the oxaloacetate, and getting all that lovely NADH and FADH2 off by supplementing that pool of succinate. And again, this occurs very specifically within a particular organelle, which we don't have as higher organisms, called the glyoxosome. And it has a particular transport system to move short-chain fatty acids in there. It has the capacity to degrade those short-chain fatty acids to become acetyl-CoA, and goes through the process of the glyoxylate cycle, with glyoxylate and 2 forms of acetyl-CoA coming in to condense in that glyoxylate cycle to produce oxaloacetate, as well as, importantly, this succinate. Now, this succinate, as I said here, has a transport system outside to move it from the glyoxosome organelle and then transport it into the mitochondria. And we'll see that succinate shuttle shortly in coming lectures. But what that allows us to do, importantly, is we get the normal process of the TCA cycle producing oxaloacetate and malate. But ideally, we can bring this out to produce phosphoenol pyruvate to make glucose from gluconeogenesis. Or we can also use our old friend, the malate shuttle. So by having now succinate coming in, so we're consuming fatty acids in the primary role to make more succinate, we're adding this to the succinate pool in the TCA cycle, the normal TCA cycle inside the mitochondria, and increasing now our malate concentrations. And with that malate concentrations, we've already illustrated the malate shuttle in regards to the movement of the malate, as well as the NADH into the cytosol from the mitochondria. And in that form, we'll make malate, oxaloacetate, go through the phosphoenol pyruvate system through and give us the phosphoenol pyruvate through the integration into gluconeogenesis to give us the sugars, to give us sucrose, or to give us the capacity to make cellulose as needed as a structural element. So again, it's an integration. requiring specific organelles to do the work. So the enzymes are contained in this particular organelle, having the capacity to access lipid bodies with a transport system and the appropriate enzymes. And then also a transport system to remove that succinate out of the glycosome to then transport it specifically into the mitochondria and then allow it to supplement and increase the pull, the normal cycle, producing malate and oxaloacetate. But now we're adding more succinate in, we're adding more intermediates, and in this case, we're going to be siphoning those intermediates off into carbohydrate production to make ideally cellulose. So again, this is plants and microorganisms mainly. So when we see this, here we have the lipid body, and this is actually from a cucumber seed. And we have the glyoxosome doing its particular job, and look at the size of it in compared to the mitochondria. So we're getting access to these fatty acids and breaking those down and transporting them into the glyoxosome, and then increasing the doing its work and consuming acetyl-CoA, but then releasing in close proximity the succinate, which will be taken up by the mitochondria, and the mitochondria then will be able to produce phosphoenolpyruvate via the malate shuttle to then get those carbohydrates. Excuse me. So again, this is more seen when we are looking at a seed in the process of germination. If you think you take a sunflower seed and you squeeze it onto some paper, you'll see the amount of oil that comes out of that sunflower seed. That sunflower seed is going to utilize that oil in the germination process. And as we know, weight by weight, there is more energy in a gram of oil than a gram of sugar. So again, it's more compact, but it needs biochemical processing to get to that ideal situation of accessing those or building those carbohydrates for germination, for cellular propagation, for leaf formation, for stem formation, root formation. That's all carbohydrate based. So again, very clear illustration of integration of metabolic pathways from utilizing fatty acids, the glyoxylate cycle, and then also utilizing the TCA cycle in combination with gluconeogenesis to provide you the carbohydrate dominance of structural materials. So again, keep that in mind. You can learn more about this if you do Dr. Owens's graduate plant biochemistry class in regards to understanding how these carbohydrates play an important role and their source in regards to energy and also cellular construction. So we had a long lecture here in regards to tracing within the TCA cycle, so you're going to have to know that. We're going to look, you've looked at the points of production of NADH and FADH2. We've breached upon the biosynthetic roles of the TCA cycle also, and we're going to come back to those when we look at amino acid degradation. We've looked at the role of oxaloacetate and that should be phosphoenolpyruvate, not PPEE, and gluconeogenesis. We've talked about the incomplete TCA cycle without going through the whole cycle and reversing the 12 o'clock to the 6 o'clock cycle, because there's reversibility, but consuming energy to make those intermediates to produce, in this case, alpha ketoglutarate. We've talked about the importance of the pyruvate carboxylase and the phosphoenolpyruvate carboxylase enzymes in how they respond to maintaining or consuming the oxaloacetate concentrations. We've looked at the importance of biotin with the pyruvate carboxylase enzyme, and as well as we've seen now the regulation of the TCA cycle in regards to the happy cell. Whether you need energy or you have surplus energy, you're going to regulate the TCA cycle to those particular needs. We've also then looked at some of the rate limiting steps of the TCA cycle. And then finally, we've examined the glyoxylate cycle in regards to the integration of fatty acid metabolism to the glyoxylate cycle its integration with the TCA cycle, and then the utilization of TCA intermediates to produce glucose moieties or carbohydrates in the forms of potentially producing cellulose for cellular plant and bacterial wall construction. So again, lots of things to think about, and you're expected to be able to work with the structures of the TCA cycle and that of the glyoxylate cycle. Okay, so we finished this particular lecture